Hello, everyone. Hope you are enjoying the content of this year's NIMAC conference. My name is RaeLynn Forsyth, and I am currently a genetics fellow at the Children's Hospital of Pittsburgh of UPMC. Prior to being in Pittsburgh, I was a genetics trainee at Johns Hopkins in Maryland, which is where I first got to be involved with NIMAC, thanks to Dr. Joanne Bedartha. And so thank you so much to the NIMAC team for inviting me this year to give this talk, which I entitled, What's Next? The Potential and Practical Aspects of Current and Emerging Treatments of Genetic Conditions. And really just hoping to give you a glimpse of where we are, where we're going in terms of treatment of genetic conditions. I have no disclosures identified by ACMG. And more specifically, while I will be referencing various treatments for genetic conditions, I'm not endorsing nor disparaging any particular treatments. Rather, in the next 30 or so minutes, I'd like to just give you examples of various treatment modalities for a variety of genetic conditions. For the sake of time and to give more of an overview, I won't be going into the scientific details of each specific treatment or the conditions themselves, but rather we'll speak broadly about treatment targets. And I think you'll see with examples that I give that early treatment through early referral is always going to be the goal, and this will involve PCPs. So I encourage you all to listen to this session's concurrent talk for novel models of PCP involvement in the referral, diagnosis, and treatment process. And before we get started, I'd just like to learn a little bit about who you are. So if you could just put in the poll, who you are, where you're coming from. And it seems we have a large percentage of genetic counselors and counseling students, some representation from patients and families, industry, which is really important to this topic, and a mix of others, which is great to give a variety of perspectives on the topic. And this talk is really just geared towards a general audience, so I hope you all find something of interest. And to get started, I just want to walk you through phenylketonuria, or PKU, as an example of where we are, where we've been, and where we're going with treatments of genetic conditions. So PKU is a disorder where the body is unable to metabolize the amino acid phenylalanine, such that in the untreated form of PKU, phenylalanine accumulates and causes neurodevelopmental symptoms. PKU is one of the more common inherited metabolic disorders with a carrier frequency in the general population of about 1 in 50, giving a prevalence of about 1 in 10,000. So still very much a rare disease, but one of the more common rare diseases. And it was in the 1950s that PKU was first recognized as a treatable cause of intellectual disability, with the observation that restricting dietary phenylalanine improved the neurodevelopmental symptoms in patients with PKU, and that those symptoms worsened again when the phenylalanine was reintroduced to the diet. Shortly after this observation, newborn screening started for the purpose of detecting PKU at birth and starting this dietary treatment before irreversible symptoms were to develop. And so dietary treatment was really the mainstay until the 2000s, when the cofactor for the PAH enzyme that is deficient in PKU was developed as a treatment. And this development coincided with more molecular testing being done, and there is correlation between genotype, that is the genetic variation in the PAH gene that causes one's PKU, and an individual's response to this cofactor treatment, such that you can often predict whether someone will respond or not to this cofactor treatment. And so with more understanding of the molecular basis for disease, so too comes more treatment options. And now a mainstay of treatment that is offered to adults with PKU is enzyme replacement therapy. So not quite a cure, even though that's where I have it listed, but I can tell you that the lab profiles of individuals on enzyme replacement that have PKU are indistinguishable from individuals without PKU, whereas those individuals with PKU on diet on cofactor often still have phenylalanine levels above that of the normal range. And now, if I can be so bold as to say, we do have potential cures for PKU coming down the pipeline in the form of gene therapy. And there's promising activity in clinical trials ongoing for gene therapy and PKU. So you'll often still hear newborn screening referred to as, quote unquote, the PKU test, which is a total misnomer, as newborn screening now includes many conditions that have treatments. And that's the biggest criteria for adding conditions to the newborn screen, that they have a treatment. Of course, the condition must be an important health problem. It has to have a cost-effective way to screen the mass population of newborns, but it must have an accepted treatment that changes the natural history of the condition. And just in my time training in the last 5 or so years, conditions have been added every year, so treatments have been developed. And beyond just metabolic conditions, treatments for a wide variety of conditions that are on the newborn screen have evolved. And I listed here these examples to show you that a variety of subspecialists are involved in treating these genetic conditions. So of this list at my institution, I may only see X-linked adrenoleukodystrophy. The hematologist will see sickle cell anemia, the pulmonologist, cystic fibrosis, the immunologist, the severe combined immunodeficiency, and the neurologist, the spinal muscular atrophy. And that just means that everyone in healthcare has to have a good understanding and access to genetics, which is, of course, one of the goals of this group. And I think I'm not alone in my belief that at some point in the future, whether it be 5, 10, 20 years from now, newborn screening is going to be sequencing, likely in the form of whole exome or whole genome sequencing, and that may be driven by emerging treatments. And there are already several studies that look at whole exome and genome sequencing as part of newborn screening. For now, it does seem like it's most sensitive when done in conjunction with the biochemical testing that's already mainstay. And if newborn screening is something you'd like to hear more about, since I'm not going to be talking too much more about it now, I encourage you to join the session at 11:30 today that we'll be talking about newborn screening and what we're doing, where we're going. But not all genetic conditions are diagnosed via newborn screening. There's often this cycle between a patient, their family, their primary care physicians, subspecialists, including genetic providers, that often is part of the diagnosis. And then once a diagnosis is made, ensuring the optimal treatment is carried through. And again, I encourage you to listen to the concurrent talk right now about ways and models of primary care physicians delivering genetic services and making this cycle run efficiently. But just to take a step back to how we often achieve a diagnosis in individuals, and that's through molecular testing, which, as I already alluded to with the example of PKU, often informs the treatment options. So starting at the top here is the most focused type of testing with single gene testing. And that's really indicated if you know of a familial variant and do familial testing for that variant, or if there's a very specific phenotype or constellation of features that points you in one single gene direction. Similarly, if you have a very specific phenotype that has a well-known set of associated genes, you may opt to do a gene panel. That is often a test that can have a quicker turnaround time, a lower cost. But when the genetic differential is broad, as it so often is, we're usually starting with exome sequencing at this point. And that is thought to cover about 85% of disease-causing variants. And I think soon too, we'll see that whole genome sequencing will become more clinically accessible. And that would also add in then the intronic regions, copy number variants, and repeat expansions. But the molecular piece is just one piece here of this puzzle of the genetic diagnosis. A genetic diagnosis also involves accurately describing clinical features that the patient is experiencing or may be at risk to experience, looking at the functional studies of the physiological consequences of the molecular changes that you may have identified, and then really keeping up with evolving knowledge of all three, the clinical findings, the molecular diagnosis, and the functional studies, so that you have a better understanding of both symptomatic and curative treatment targets. And when we're talking about treatments for genetic conditions, we can divide them based on where they fall into the central dogma of molecular biology, where their action takes place. And remember that simplistically, DNA is transcribed into RNA that then is translated into an amino acid chain that is folded into a protein. Often these proteins are enzymes that require substrates to then go on to produce a product. So treatments can actually target DNA, RNA, protein, or the substrate. When we think about protein-based and substrate-based treatments, that's targeting the downstream consequences of a genetic mutation, whereas DNA and RNA-based treatments target the actual pathologic change itself. And so in the next several slides, I'll highlight several examples of each of these treatment targets and the conditions currently utilizing such treatments. And again, I'm not going to go into much of the detail on any particular treatment or condition, but rather give an overview to show the breadth of application. And so probably the most well-known genetic treatment strategy are those that are substrate-based. And these are what I use every day as a biochemical fellow. They're the mainstay treatment in inborn errors of metabolism, such as the example I gave you with PKU. So you can restrict or avoid an offending substrate, such as through dietary therapy, or you can remove a toxic substrate that is formed. In some disorders, such as biotinidase deficiency or congenital hypothyroidism, you actually have to give back or replace deficient metabolites. But going back to some of these other examples here I have listed, we can also think about tailoring treatments in pharmacogenetics in this way, such that we may adjust doses of a medication, in this case, the medication being the substrate, based on if someone is a slow or fast metabolizer, perhaps. I also wanted to list tyrosinemia separately here because the mainstay of treatment in this condition is using a medication called nitisinone. Now, nitisinone works by preventing the accumulation of toxic metabolite called succinylacetone. That's really not important here, but I just wanted to point out that nitisinone, it's interesting to note that it was first developed as an herbicide and then only later discovered to be a proven treatment for tyrosinemia. And so that leads me here to this last example of thinking about novel uses for already existing medications or drugs or substrates. And that is, in this case, I'm saying using a small molecule to block a receptor, such as in receptor antagonism, where in Marfan, this has really been shown to be helpful, and that the common antihypertensive medication, losartan, an angiotensin receptor blocker, helps to ameliorate the TGF-beta pathway abnormalities in this condition. But just thinking broadly of that as an example, I think we'll see that more common medications or things that have already been out in the world for other uses can be repurposed and discovered to have new roles in rare disease treatment. And that's through a process, perhaps by small molecule screening. So this is a schematic showing high throughput small molecule screening, where already approved drugs are virtually screened with a variety of methods to see if they have drug-like properties for a particular target that an assay has been developed for. If they do, it's developed then as a treatment for that condition, put into clinical trials, and if those are successful, it may eventually attain FDA approval for that indication. And so I think that's maybe how we get more substrate and protein-based treatments, is just looking for things that are already out there in the world to see if they have novel uses. When we think about protein-based treatments, then those again are largely a mainstay of treatment in inborn errors of metabolism, where you can replace a deficient enzyme, such as in the case of lysosomal storage disorders, but also in things like hemophilia, where you treat by giving the deficient clotting factors. I'm going to skip organ and cell transplant for a minute, but then go to cofactor supplementation. So if your deficient enzyme has a cofactor, supplementation with that cofactor may also prove to be beneficial. Again, I used PKU as an example there. But clinical features of several disorders have now been shown to be prevented or attenuated by transplant. And so, for example, X-linked adrenoleukodystrophy, Krabbe disease, some of the mucopolysaccharidoses are some of the conditions that have recently been added to several states' newborn screens because bone marrow transplant, when offered early, has been shown to either ameliorate or prevent or attenuate the symptoms of those conditions greatly. Liver transplant, something I have to mention being from Pittsburgh, is also done more routinely now for several indications. Both of these procedures, bone marrow transplant, liver transplant, of course, not to be taken lightly. They are big procedures, but they do provide a permanent supply of deficient enzymes. And so when you think about something like a urea cycle disorder, where an individual without a liver transplant is kind of constantly at risk for hyperammonemia and the devastating neurological effects of that, after a liver transplant, they are really considered cured from that condition. And lastly, in this category of protein-based treatments is chaperone therapy, that is kind of helping proteins work better. And so also being further developed for various conditions. And cystic fibrosis is probably one of the best examples here where chaperone therapies are being used. And again, you have to kind of go back to the molecular genetics to understand the consequences of the DNA change and why this therapy would work. And so the most common mutation in cystic fibrosis, F508del, causes the CFTR protein to fold incorrectly. And that misfolding is amenable to chaperone therapy, as is the faulty or deficient CFTR functioning caused by some of the other mutations. If you don't even make CFTR protein though, because of a nonsense mutation perhaps, then you have to think about RNA-based strategies. And so that leads us to those strategies here. So in the case of nonsense mutations, like we just saw in cystic fibrosis, translational read-through agents can actually enable protein products to be formed and then used correctly. In some of the hematological disorders, however, though, we just kind of bypass where the gene expression is faulty and go elsewhere in the DNA code in the RNA and upregulate gene expression elsewhere, such that you then produce a protein that can counteract pathologic or deficient proteins. So in sickle cell anemia, beta thalassemia, you can actually upregulate protective or helpful hemoglobin F production by means of various treatments. Altering gene expression is also the goal behind antisense oligonucleotide therapy. And these therapies either act to upregulate RNA that is transcribed into beneficial protein products or remove RNA that leads to toxic protein products. So the celebrity here of this group right now is the antisense therapy for spinal muscular atrophy, which allows for increased production of the deficient SMN protein by upregulating SMN2 transcripts. So some of these other disorders here are still in clinical trials, but again, one of these therapies that is kind of, going to be a wave in the future of treatment strategies. RNA interference is also being studied, and especially in more complex conditions like cancer, so not just the monogenic diseases that we tend to think about in our world. And so I previously discussed really how common medications may be used to treat genetic conditions, but in this case, a genetic-based treatment strategy is also being used for common conditions. And I do think once we have a better understanding of the genetic basis of common complex diseases, this will be happening even more. And last, but certainly not least, is gene therapy. That is the direct alteration of the pathologic DNA change. And this can be done by similar kind of goals as the RNA-based treatment. So you can replace a gene such that an essential protein that was deficient is now produced. Or you can inhibit a gene similar to interfering at the RNA level, such as in the case of toxic overexpression. So you inhibit that gene so that the damaging protein is not produced. And I think many people probably have heard of CRISPR, a form of gene editing, where the problematic section of a gene is corrected. And lastly, you can also add a gene to then produce a protective protein, similar as to how I described before with the hemoglobin F production. So if you look for gene therapies that are FDA approved or approved by the EMA, the European equivalent to the FDA, it's only a few right now. Here in the US, we have FDA approved gene therapy for a specific type of retinal dystrophy and spinal muscular atrophy. This gene therapy for spinal muscular atrophy is different than the RNA-based treatment that was already really revolutionizing the treatment in this condition. So two really new kind of very good therapies for that. And gene therapy is also approved for several cancers. There was a gene therapy for lipoprotein lipase deficiency, but that's no longer on the market, really just because the expense to keep it on the market was too great for the demand. So it's something to think about with gene therapy is the cost, but I think we'll see that just like many things in genetics, like the cost for these treatments will go down eventually. But there is an increasing number of gene therapies in development, and including at my institution, which is kind of exciting. So if you would kind of in your own mind think just for a moment, how many clinical trials do you think are ongoing for gene therapy? Just put them in the chat. And it seems to be a big range coming in, kind of like tens, hundreds, thousands. And so this is a graphic from a resource that I liked when I was preparing this talk. It's called genetherapynetwork.com. It's a website whose mission is to, quote unquote, provide healthcare professionals with high quality scientific information about gene therapy. And they really do have good videos, good graphics. And this is one where they show that now we're really at about 200 new clinical trials approved per year. And that number does seem to be climbing, especially since like kind of the 2010 mark. And also increasing, maybe even more dramatically, is the number of investigational new drug applications for gene therapy. And an IND, the investigational new drug application, is the step before administration to humans. So again, right at that 200 mark. And so what I do now before I see a patient is look at clinicaltrials.gov, see what clinical trials that are there for that diagnosis, where they're at, what phase they're in, if it's something my patient would benefit from, would be eligible for. And so if you just go to clinicaltrials.gov, search for gene therapy, those trials that are recruiting to give an intervention, that number was 839 when I made this talk, likely to be different, probably even more the next time you check. And so of those, most still are in phase 1 to evaluate for safety, but there is a sliver here of phase 3 to confirm effectiveness and some in between, of course. Many of the trials are for various cancers, but again, a sizable chunk here for monogenic diseases, which is what we kind of typically think about for genetic conditions. And so these diagrams here then just summarize all of the treatment targets from DNA to protein with the examples as I had listed. These are from the most recent edition of Thompson and Thompson, one of kind of the hallmark textbooks in genetics. And so I'm not going to read through again all of these, but just some good graphics to walk you through DNA to protein-based treatments and some examples of the conditions. And then just on the right, I wanted to point out that we, of course, still have to think about the clinical phenotype of our patients and treating symptoms as we would in any other case. So with the various subspecialists being involved in the care for the variety of symptoms that a patient can present with or be at risk for. And then also treating the family as a unit. So even if we might have a curative treatment for one individual, that there's still a family then that is potentially affected with a condition. And so offering genetic counseling, carrier screening, pre-symptomatic diagnosis, and other therapies for that family unit. There are certainly challenges and considerations to be had for any treatment, which I didn't go into for each for the sake of time. But just speaking broadly, of course, the earlier the treatment, the better the outcome. So obtaining an accurate and early diagnosis is important and often relies on really a high degree of suspicion by PCPs, other subspecialists, and expanded access, all goals of NIMAC in this group. Any treatment can, of course, have adverse effects, and I would be remiss without mentioning that the progress of gene therapy really was stalled with the tragic death of a study participant. Jesse Gelsinger was in the early gene therapy trials for OTC deficiency and died. With that did come more safety protocols, and rightly so. The access to early trials is limited to experienced providers and care centers. And it just really does then limit, who can be in these trials and where they are. And as genetic conditions become more treatable, as we're doing a better job at offering treatments and helping individuals live longer, we're then learning of new features that may require attention and treatments. So really, the work is never done. The work is never over. We need more people to do it, always. But I did find this hopeful and promising. So Figure 1 is an online forum for all medical professionals, not just those in genetics. It was originally created for people to kind of crowdsource input on cases where they had medical images that could stir some conversation. And now really has become this educational community. They have quizzes, they have podcasts, CME opportunities. And just by chance, when I was making this talk, the current podcast was all about gene therapy. So they do have this kind of dramatic quote from one of the episodes. Just 40 years ago, most genetic diseases were thought to be untreatable. They've been seen as a permanent part of the human condition. But today, we are tantalizingly close to revolutionizing not just genetic disease, but cancer and even aging. So maybe a little bit of a hyperbole at this stage, but nonetheless, I think getting our non-genetic colleagues interested, excited, thinking about, talking about genetics, both in the rare and the common conditions. And so thank you so much for listening. I hope you found that framework helpful and are equally excited about where we're heading. These are some of the resources I had mentioned, and please just reach out with any questions you may have, or if you'd like to discuss anything further. And I hope you enjoy the rest of the conference. Take care.
And thank you, someone who's already right on that, sharing what they learned. I'm gonna put the link in chat here. If you're just joining, this is how to participate. What's going on is go ahead and click the link in chat and answer that question. All right, we're seeing answers coming in as Kenzie's opening the door. I think we have people still coming in the front door. I'm going to keep pasting in chat because those of you who are in the session, you see all the stuff happening over and over again in chat. And that's because when you join a new Zoom meeting, when you're just coming back, it's like a clean slate. And so the people who are just joining can't see what was pasted before. All right, Joanne, should I put you up here to speak to what you're seeing coming in? Go ahead and unmute yourself, and then I'll be able to hear.
Group is mostly here and really reinforces the themes that our speakers so thoughtfully generated around thinking of new models, working on genetic counseling billing, and really trying to shorten the the trajectory from genetics to primary care. And again, I think that's what these models are under this group. Do you have any sense of when the other group is going to be coming in? Are they here?
They're coming right now. I just heard a notification in the back channel.
Yeah, no, I think again, the we really people are so good about, I think, great listening to great speakers here is what we I'll repeat, the need for genetics in primary care is now as maybe the theme for our session.
Yes.
I think just in the interest of time, I know, how do you want to do this? Do you want to, we're up to 111. I don't remember. I think we were at 119 when we started.
Yep, we can see there's still people coming in the waiting room. Okay. And so just when that slows down, I'll give you a cue. And I'm pasting in chat here so that people who are just joining have a chance to participate in this because they can tell something's going on.
Yeah, and when then, I guess I saw that I was getting different, where I got a couple messages about how long we have now, I guess, till the break.
Yes, we will go to the break in just 5 minutes here.
Okay. All right, well, maybe the folks can be, the speakers can be transitioning again to the question of, what are you doing now that you can see replicated in another center most quickly? And I use December 2022 as a goal for that, but what would be those quick steps to try to make some of these ideas happen? So that's for Linda, Alex, and RaeLynn.
Okay, great. Kenzie, can you bring those folks up to the stage if they are in the meeting?
Yes, maybe not all of them are here yet, but we've definitely got at least one.
I got Alex there.
I'm here.
Oh, hey, RaeLynn.
Hi.
And then Linda, maybe we can see if we can.
Yes, everyone's here now. Great.
Okay. So again, we have just a few minutes now, and appreciate, what I know was a great talk from RaeLynn, and I heard Alex and Linda, but I'm going to ask and try to pay attention to the chat. Of all the things you talked about by way of improving access and models and amazing funding and bringing people in and teamwork and collaboration, is there one thing you could say, and I'll say a genetics professional or a family might have to partner to start this, but what's one thing you're doing that you think could be done this year in another center? And maybe I'll start with you, Alex. I don't know exactly how long it took Carol to recruit you and train you and all the rest, but you think your model is reproducible in a center now?
Yeah, I do think so. A lot of the Linda went through some of the different models. I do think the hub and spoke model is the easiest to, and that means outside of the medical home, a separate clinic of a primary care provider and a genetic counselor together. That kind of model the benefit of it is it can be replicated fairly quickly. There wasn't a a huge amount of training that I needed beforehand. Most of it I received during my residency. And so that was something that we brought about in a few months, and it has been successful. I think also having the genetic counselors there is really a big part of it, that they're just so knowledgeable and can really help me guide me through this process.
Great, thank you, Alex. Linda, how about you? If you could choose one thing that's the fastest to replicate, and I mean, obviously in a thoughtful, collaborative way.
Yeah, I think the very first step is to identify a primary care physician champion, whether they have the background or not, as long as they have the passion and the interest, and then in parallel finding those genetic counselors who would be excited to take on this primary care challenge, and partnering those groups together to begin the discussion and investigate how they might be able to launch into creating that hub and spoke model.
RaeLynn, can you hear us? Thank you, Linda.
Yes, I can.
Great.
So I didn't talk too much. Oh, is my prompt the same?
Well, your prompt is the same, but I was going to say, if you wanted to, because I know of your slides, if you wanted to use PKU as an example, because that's certainly one where, again, there is a lot of back and forth with a PCP potentially, but your call on how broad you want to make your response.
Yeah, so I did talk about PK, since you brought it up, I did talk about PKU to our group as kind of the hallmark condition that we have that has been amenable to treatment, and we've seen treatment evolve. And so I think it is one of the more common conditions too. So I also just talked about kind of this cycle of communication of the PCPs working with the subspecialists. I don't think, PKU in general is a great example, but just in general, people kind of recognizing that there's evolving treatments for various genetic conditions beyond just kind of the ones that we think about in terms of the genetics clinic that we see, but sickle cell, cystic fibrosis, amyloidosis, some of the neurological conditions like Parkinson's, ALS, are all having evolving therapies. And they're in the news and they're on podcasts. And so to kind of empower our colleagues to check those things out and be excited about those things, so they can be part of the team here.
And maybe one last comment I will take from that. Thank you so much. Linda, any thoughts ahead on partnerships that involve nutritionists or, folks who can follow up on the pharmacogenomic as it moves to potentially, broader issues, or how you're thinking about expanding the model for other professions where there may be a shortage of people, and yet we need to figure out the distribution of service to make it more accessible for families.
Yeah, so we actually do have a nutritionist who is a part of our team as well. Sorry, there was a lot in that question, so I'm trying to just.
No, I'm thinking on my feet or somewhere between my ears. I guess I was asking. I think that the challenge is the individuals who take the most time, in a sense, and almost require a one-on-one precision thing. I don't think any of us entirely have our heads around how that can be in a distributive model. On the other hand, those folks are often closer to home, et cetera, and how we think about, particularly for things where, we're covering almost 1,000 metabolic patients here out of Hopkins. How do we think about those models when we start to add to, as RaeLynn has talked about, so many more diseases, so many more therapies, but are there some that could be handled in another way of thinking about collaborative care with primary folks at the center, in a sense, or with another team member?
Yeah, so I think there, right, the key is communication, right, and opening up the lines of communication between the primary care providers and the subspecialty providers, and especially around complex management of these underlying syndromes or metabolic conditions, and really providing some guidelines for primary care. I think, when most of us trained in primary care, we got to something genetic or something metabolic, and we sort of had this reflex to refer. And we need to evolve our thinking beyond that says we can actually co-manage patients, right? And this concept of co-management can give the primary care provider the support they feel like they may need if they have questions or concerns to be able to reach out to the subspecialist without overburdening the subspecialist to actually be the primary manager of all of these patients. So I think it's that co-management model is really important. I also think that there's a scalability issue here where we need to, Alex and I need to help everyone else recreate these models at other institutions. And I think we'd be very happy if anyone has questions to reach out to us. But one of the things that we're doing here is we just actually launched a training track in our family medicine residency. And so we have our first resident that matched this last year, and he'll be doing his second and third year continuity clinics with us in our precision medicine clinic to actually start thinking about how can we scale, how can we create more trained providers if we do go with that hub and spoke model. So I think it's the co-management and thinking more about scalability.
And there are not, to my knowledge, family medicine genetics combined residencies. Is that something you see in the future?
Correct. There are not as of yet. I do think that's something we really, really need to think about is that primary care genetics combined residency with the intent to get the provider back into that primary care facing space as opposed to subspecialty medical genetics facing space.
In full knowledge that people who need a break should take it, I'm going to continue here for a moment, Erica, and all your independent people. And if you need a break, please take it. Alyssa asked here, particularly to Linda and Alex, again, if there are specific things that NIMAC could do to grab the interest, engage, and collaborate with family medicine and the primary care community.
Alex, on that one?
Sure.
I'm sorry, I didn't hear what you said.
Do you want to share any thoughts on that, or?
I'm not sure exactly. There's nothing specific that's coming to me on that one. I'm not exactly sure the type of services that can be offered. It sounds like a great idea, but I can't comment on the specifics of that.
Linda?
Yeah, I'm wondering if we could just start with an interest group, right? That connects and shares discussion and ideas and possibly meets occasionally to think about how do we scale and expand this type of collaborative effort.
Yeah, I'll just offer up, there's another comment in the chat about genetic nurses, and some of you may know we've had some interest in the nursing school, had a planning curriculum, community of practice. Nursing school at Maryland was involved in some of the community places. The challenge right now, and we had somebody talking yesterday in West Virginia, that the paying for that training and certification, which can involve multiple years of working with clinics. So one thing we do is try to be open to having DNPs and advanced practice nurses rotate through genetics, and if they are particularly interested in specific conditions, support them through community of practice in the nursing school that has other people in it. Between Erica, Alyssa, and I'm not getting more phone calls here. I'm looking at my phone. I don't have any urgent message. RaeLynn, did you want to say anything? And then I'm willing to told we need to.
I just wanted to comment on a couple of things in the chat because I think that there are really good ideas here. Is to Jessica mentioned to use genetic nurses in genetics as an option, and so utilizing all of our healthcare providers in creative ways, that it doesn't just have to be an MD/DO that's overseeing these things. Again, pushing interest. And then somebody commented on creating a complex care rotation within pediatric residency, which Joanne, is near and dear to our hearts, is training the next generation, right? And so I think from a NIMAC perspective, to approach some of these educational bodies on, incorporating genetics training within training programs for other subspecialists. I think they would be very well received by trainees. That's what we've demonstrated, and are important.
So I think I've now broken all rules here, but that's okay. So there are lots of other things that suggest connections here. So for people who are particularly interested in any of the topics that have been addressed here, there's a suggestion also about telehealth issues. And again, that's something that we have session later. So maybe I'll ask Alyssa to make sure that, we come back to this part of the conversation in that. Erica, what happens to people who break rules?
We all support them, Joanne.
I see. All right. Well,
Their judgment in the moment.
all right. Well, I really think this has been a wonderful session. I'm sorry for whatever technical glitches occurred for people, but let's keep these conversations going and all. And I think people now have a 2-minute break. And then next comes the collaboration. Is that not correct?
Yes, that is right. And I'll go ahead and put a sneak preview in chat of these are the vendors.